Chapter 25, Part 2, Narrated Lecture. So looking at here, the catabolism or breaking down amino acid, where you're going to start with the removal of the amino group, and once the NH2 is removed, well, it's going to go into either synthesis of new nitrogen compounds, and we're going to look at those and the way passage of nitrogen can get into the urea cycle, or incorporation of the carbon atom into compound that can sometimes help to increase the amount of energy through the citric acid cycle. So our bodies do not store nitrogen containing compound, and ammonia, NH3, is highly toxic. So they have to just get removed. So there are some different paths. The amino nitrogen from the protein can go two different ways. It can either be incorporated into urea to be excreted, or it can be used in the synthesis of new nitrogen-containing compounds. Those are here nitric oxide, NO, that's a chemical messenger, or hormone, neurotransmitters, a coenzyme found in nicotinamide, heme, which is part of the hemoglobin that you find in the erythrocyte or the red blood cells, and also for the nucleic acids such as the making of the bases purine and pyrimidine. So let's just go back to nitrogen oxide, NO. That's a very interesting molecule because it's a free radical and it's very reactive. Helps to lower blood pressure, kill invading bacteria, as well as enhancing memory. It's synthesized from oxygen and the amino acid arginine. In the blood vessel, nitrous oxide activates reaction in smooth muscle cells and help to the dilation, and helping therefore to decrease the blood pressure. Now looking at the carbon atom of the amino acid, where they can be converted into compound that can go inside the citric acid cycle. They are going to keep going through that citric acid cycle to help making energy and carbon dioxide. About 10 to 20% of our energy is produced this way. If it's not needed immediately for energy, the carbon carrying intermediate can go into storage, such as triacylglycerols, glycogen, or even the ketone bodies. So looking here, the first step in the amino acid catabolism is to remove the amino group, and that can take place here in the cytosol of the liver cells. Now, I'm expunging a lot of the reaction. You just need to know what is transamination. Well, transamination means that the amino group of the amino acid and the keto group of the alpha keto acid are going to change and switch places. If you see here, amino acid 1, alpha keto acid 1, there is an enzyme called the alpha transaminase. It's going to just replace those, so you get the alpha keto acid 2 and another amino acid number 2. So that's a key reaction for many biochemical pathways. Those reactions are reversible, which is indicated by the double arrows. One goes to the right, the other one to the left, showing it can go both ways. And amino acid concentration are regulated by keeping synthesis and the breakdown in balance, which is what I have summarized right here. Another point important is called oxidative deamination. This is where you convert amino acid NH2 into an alpha keto group, and the way you do this is removing an ammonium ion NH4+. Glutamate from transamination serves as an amino group carrier. Glutamate can be used to provide amino group for synthesis of new amino acids, the ones that are non-essential, but most of the glutamate formed in this way will be recycled into generating alpha glutarate. And then the last slide here, just looking at here, the ammonium ion formed in this reaction proceeds to either the urea cycle, where it is eliminated in the urine as urea. Just look at here, just that cycle, just understand that alpha amino acid starting there and ending here into urea.